Hi there, guys. This is our YouTube channel Biotech Amalgamated. Today, we are going to discuss on the topic of AMES test. We live in a world that is being industrializing for quite a while. The food industry similarly wants to cope up with it. To cope up with industrialization, it uses a lot of chemicals. It adds a lot of food preservatives, food additives, flavoring agents in our foods. Those are basically chemicals. We consume them unknowingly. We don't know that whether they are beneficial or harmful to us. Some of those chemicals might be mutagenic, or they can be called as mutagens. Mutagens are those that help substance to convert into carcinogen or might cause cancerous effects. So this test was made by Bruce Ames to detect whether a chemical is a mutagen or not. So uh, this test was developed by Bruce Ames in 1970s. It follows the principle of reverse mutation or back mutation. I'll tell you about back mutation later. Let's proceed with the protocol of the test. Now, what Bruce Ames did was that he took a strain of Salmonella typhimurium that was actually defective. Defective means that it couldn't produce a single amino acid, histidine, because of a defective gene, but it could gain the ability to uh, synthesize histidine with the help of some kind of supplemented chemical. This thing is known as back mutation. Like, suppose you are giving a little bit of chemical that causes a change in the genome of the salmonella, of the bacteria or any species, which helps the uh, organism to synthesize a particular uh, protein that it couldn't synthesize earlier. So now these type of strains are also known as auxotrophic mutant. That is, they can uh, gain the ability to synthesize a particular amino acid in the presence of some kind of chemical. Now let's look at the protocol. Let me take a pointer to explain it properly. Now what Bruce Ames did was that he took a salmonella strain, an inoculum of salmonella strain, which is His negative. That means it couldn't produce histidine by itself, but can produce histidine with the help of some kind of mutation caused by a possible mutagen, a possible chemical. So, along with it, into the inoculum, rat liver extract was added. This was added in order to increase the uh, rate of production of Salmonella strain. Now, the protocol was divided into two different sub-protocols. Like in the first one, the test protocol was done. And in the second type, only control protocol was done. Only difference was that in the test protocol, the possible mutagen was added. The possible mutagen or chemical that was added was 2-aminofluorine. Now the inoculum was plated in a media that contained a little amount of histidine, and both the plates, control and the test plates, both of the petri dishes were incubated at 37 degrees Celsius for 24 hours. Now what the results were obtained was that it was obtained that in the test petri dish, a huge amount of salmonella colonies were obtained, whereas in the control plate there was a little amount of salmonella colonies. Now what Bruce Ames inferred from these results were that the chemical that was added in our test protocol caused a mutation in the genome of Salmonella typhimurium, and that helped in gaining the ability to synthesize histidine through back mutation, whereas that was not possible in the control plate. The little amount of colony that was obtained in the control plate was due to the minimal amount of histidine added to it. Now let's look at some of the applications of AMES test. Now, I told you earlier that the food industry is adding a lot of flavoring agents or food supplements, food additives, food preservatives in order to increase the deliciousness of the food. It was found that a flavoring agent like saffron was found to be carcinogenic with the help of AMES test. Similarly, there are various drugs that we uh, take, like medicines, like isoniazid is an anti-tuberculosis drug that was found to be mutagenic. Now, as I told you earlier, that the food industry adds a lot of food flavoring agents, food preservatives in order to increase the shelf life of the food and as well as increase the deliciousness of the food. One such chemical that is added in the form of food preservative is sodium nitrate. It is not initially a mutagen, but the moment it comes in contact with the HCl present in our gut, it converts into a potent mutagen. It converts into nitrous oxide through biotransformation process, which is actually a carcinogen. It also helps in converting a defective gene of a particular strain, of a particular bacterial strain, into a functional gene. Like in the previous protocol, we saw that Salmonella typhimurium could not produce histidine by itself because of a defective gene. But the moment it got mutated, it converted the defective gene into a functional gene. And by that, it was able to synthesize histidine by the supplementation of a chemical. That's all for today's topic. I hope you liked it. Thank you.